Welcome to Genetics at City. In this video, we are going to discuss about some other important concept for the SRF genetics and plant feeding. Yesterday, we have discussed about some important topics that may come for examinations or may not be, but it's very important for the examination point of view. And today also, we are going to discuss about some more concepts about uh, the some more concepts which are related for the SRF genetics and plant breeding. And the first and most important concept is gene pool concept. I think many of you are very well aware of this topic because you are reading from the UG days, but you are going to miss some important points about gene pool concept. First, you should understand why we created a gene pool concept, why Harlan and David actually created this concept. It actually, these gene pools are not made to change any taxonomic order of the species. It not means like in GP1 there are different species are present. It's not saying like this particular GP1 group is a separate taxonomic group. It's nowhere related to the word taxonomy. You are not changing any taxonomic concept. It is just made for the breeder convenience. For example, if the two species are easily crossable or not. Okay. Directly, there is a question were asked. Gene pool concept were gene pools were created based on the convenience of crossing, and there is some other points also. Generally, they will try to confuse you, but if you know the core point about this concept, you will not never going to confuse. So we should remember it's not made to change any taxonomic order. It just created for the convenience of the breeder. Either the two species are easy to cross, then put them in GP1. It is very difficult to cross, put them in GP2. And many times if they will confuse you. For example, if they need progenitor species they have given, and if they ask which is the uh, either this particular progenitor present in GP1, GP2, or GP3. Without any doubt, you should keep an option like GP1 only. For example, if any progenitor species like Sisal reticulatum, they ask it is present in GP1 or GP2 or GP3. Without any doubt, you can opt the option GP1 because all the progenitors will be present in GP1 itself. And you should also remember what are the important species present in the gene pools of wheat, rice, maize. For example, Elymus, Lamus, and there are several others who are important species present in the GP3 of wheat and their economic importance. For example, biological nitrification inhibitor gene that present in the Lamus species of the wheat. And in rice also, last time they were asked about Vareza nivara was identified from the state of India. And there is another question like coigs species are present in the which gene pool of the maize. So these are the different, and Vareza diperennis was discovered by whom and what is the importance of it. Like that, there are several questions are repeatedly asked in the previous examination. That's why you should thoroughly go through about wheat, rice, maize, and I made a clear video about all these gene pool concept and important aspect of these crops, and it's present in my channel. You can watch the videos. Then you should also remember that four different complex of the rice. That is also very very important. Then another important concept is pre-breeding. Many of you know already about what is pre-breeding, but you should also understand the core concept of this pre-breeding. Mainly, what is the why we are doing and what is the use of the pre-breeding is very very important. And who given the term? The Rick given the term, and majorly we are using to create intermediate lines that can be used for the further breeding program. So act as a link between the gene banks and the breeding programs. Then another important concept is center of diversity. And many will stick to BDC chapters only, but it's not like that. You have to read from different sources. You have to read this topic from the different sources. For example, last year there one question was asked about this the crops which are derived from the progenitor with a wide dispersal and frequently involved in multiple domestications. Which one is either it is endemic or monocentric, non-centric or oligocentric? See, generally when you have a multiple domestication sense, when particularly they should not have any one side of center of origin. That type of plants are generally called as non-centric crops. And there are examples also present in the Chahal and Ghoshal book. You should read this concepts before going for the examination. Then another important concept last year repeatedly they were asked about Fisher's fundamental theorem of natural selection. Like earlier, there was one question called as that evolution is the product of. It's a product of selection pressure and the your genetic variance in the population. By looking at the option only, we can able to tell the answer if you know the core concept. If you don't know the core concept, it's very difficult to answer. For example, if the Fisher fundamental theorem of natural selection says that the average fitness of the population is mainly due to the natural selection that is equal to the genetic variance in the fitness. The high the genetic variance present in the particular species, then definitely they have high fitness. And it is also related to the selection coefficient. It is also related to the genetic load in the population. And these are all the concepts are linked to each other. What is genetic load and what is genetic polymorphism and what is polymorphism and what is balanced polymorphism? What is some there are two concepts. One is permanently present and one is transient polymorphism. And transient polymorphism due to which phenomena and balanced polymorphism due to which phenomena and how polymorphism is maintained in the population. If there is a polymorphism, definitely there will be high genetic variance. So these are the concepts are interlinked. It's clearly explained in the Faulkner book. If you read the Faulkner book, you will going to understand very easily these concepts. Then another important theories along with this recently asked and it may going to ask like Fisher infantism model. If you read this paper, you're going to understand. But you just type Fisher infantism model in ChatGPT, you'll get some information that is more than enough for the examination. And another concept called as central limit theorem. Another important concept is neutral theory of molecular evolution. For example, what are the neutral? What is the theory mainly explains about what is the role of neutral mutations that's not important for the phenotype development and how it is maintained during the evolution and how it related to the evolution. It's very interesting and it's very important for the plant breeding aspect also. Like today's marker that we are using, there are one of the related concepts to this particular theory. And there are another concept, inbreeding coefficient, Balmer effect, and repeatability. And these are all the concepts are present in the J.R. Sharma book and Faulkner book. If you just type, otherwise, if you don't have time now, you just type these words in the ChatGPT or any other or in Google, you'll get some information that is more than important, so more than enough for the examination. Then, for example, some easy question that you should answer by your common sense, by using your common sense. For example, you see this agrobiodiversity is increasingly threatened mainly due to introduction of modern crop varieties, overexploitation of natural resources, changing the food system and social change. See, by looking at three options, you can easily guess A, B, C will be right answer. But the question arises either the lack of excessive conservation infrastructure will lead to loss of agrobiodiversity. It's not like it is only to conserve. It will not threaten the uh, agrobiodiversity loss. Okay, that's it's a kind of a reasoning question. So you should, by using your common sense, you should answer this type of question. That's why you should be very attentive. And one important thing which I want to tell you all my juniors is you should be very attentive in the examination. How much you read is not important in the examination, but how much you are attentive in the examination is very important to score high. Then another important concept like biodiversity, you can see by looking at the option, it gives them positive feeling that all options were correct. Actually, the all options were correct. Like this, there will be a lot of questions. Then don't miss these type of questions because everybody will answer this, and most of the time it will be chances of getting right will be very high. Then another important aspect like conventional biodiversity, and anybody will read just when it is came and why it is came. Sorry, anybody will read about mug up these points like they will mug up the year, they will mug up the some important points related to the CBD, but nobody will going to understand the core principles of the CBD. The core principle of CBD is tells about earlier the plant genetic resources they thought like it's a like a heritage of the mankind. So, if for example, if the germplasm present in India, the European people can easily use without any permission from the Indians. And if the Indian want to use any germplasm present in Africa or anywhere, he can you directly use because it's a God gifted, it's a uh, God gifted boon like that you can think. But uh, this is your earlier concept. But what CBD tells, every country have their own sovereign right over over their genetic resources. If any country want to use your country resource, then they should take the permission from you. And what are the benefits are arising from the using this germplasm? They had equally shareable. They had to share equally like profit of sharing, like benefit of sharing and sovereign right over the plant genetic resources for their country. These are the core principles. But USA don't want to. USA against for this principle. That's why they are not ratified for this CBT. And there are some three other more countries also not ratified, but not ratified for this treaty. And you should also read about this. And it's very, very important for the examination. And there are several protocols and present under the CBD, and like Nagoya Protocol, Cartagena Protocol, and CBD years. And many times they will ask about the concept, like CO COP, COP, recent COP where it is happened, and the conference of party meetings. Generally, like there are two COPs are there. One is under the CBD, one is under the like United Nations Environment and Climate Change related COPs are present. Generally, feel confused. Like CBD still, I think in 13 or 14 COPs are going on, and in that UNCCFF, there is a like 32, 33 COPs are present. So you should not confuse about the COPs of CBD and COPs of the United Nations climate change aspect. So that's why you should read about recent COPs, and we should also read about the countries ratified for CBD, which countries are not ratified CBD, core principles of the CBD. It's very important, and protocols and year is also very important. And there, this last year they asked chronological order of the protocols under CBD. So it's very, very important for the examination point of view. Then, for example, there is another most important thing we're asked like about normal distribution. And I suggest you to read the properties of normal distributions very carefully. Each and every property is very important while deriving your any quantitative genetics concepts. That's why reading the properties of normal distribution is very, very important. They have directly asked, for example, if the negative cumulative normal distribution, it emphasizes the extreme values in the distribution. For example, if there is extreme values are represented by the negative cumulative normal distribution. Then there are several tests to test either your data set are following normal distribution or not. This is also a very important aspect for the examination. Generally, we use Shapiro-Wilkes test and Anderson-Darling test and histogram test and QQ plot analysis to test either the data present in the normality. If the data is not present in the normality, you can do analysis. But there is another way. You can transform the data. There are several ways to transform the data, like arc-sign transformation, lock transformation. You should know about which are the in which case which transformation method is used, and which tests of normality is better. And there are different cases are present. You should aware of all these concepts for the examination. Then then we should also read about what are the important quarantine laws and the chronology also important. And there are last year there was a question asked about the quarantine test of in India. It's like Colorado potato beetle as the answer. There are some like for example the rice potato tuber was introduced from which country. Regularly there are some questions were asked, and please refer the training manual present in the NBPJ website for these particular concept. Especially read the quarantine related chapter, and initial chapters are very, very important for the examination. Then there is another concept like ecology and evolution. Read Pranakumar book. Read basic concept like the different indices used to measure the diversity in the ecology, like Shannon Weaver index, Majorly, it is measure the phenotypic diversity. And if you don't know also, by looking at the option only, you can guess. For example, if the term used for the genetic related, generally it's a kinship. Then assess the marker polymorphism is polymorphic information content and determine the mutation is nowhere related to Shannon-Weaver index. Generally, the third option would be the correct. That elimination also very important while answering the question. But to eliminate, you should also know about these concepts also. But anyhow, you are knowing about what is polymorphic information content, what is kinship, and what is mutation in the population genetics. Then easy to eliminate all these options, and you can able to opt a right option. Then the another concept called a species concept. There are different species concept and who given this species concept also very important for the examination point of view. There are many questions were asked previously and definitely there will be one question from the species concept. Along with the species concept, there is another concept called a speciation concept like allopatric speciation example, parapatric speciation example, sympatric speciation example, and hybrid speciation example. These are very important for the examination. Then there is another concept called as ITP GRFA. Here also USA is not the ratified for this ITP GRFA. When it came and Annexure 1, which crops are present, which crops are not present, and why the treaty came, and even presence of CBD, what is the necessity of creating another treaty for another convention for this? Then you should read all these aspects before going for the exam. Then the another important concepts are test of significance. Read ICR notes for this to refer this concept. For example, what is test of significance? What is null hypothesis? What is alternative hypothesis? Type 1 error, type 2 error. Last time they asked about type 3 error. And why we are using these errors in our experimental designs? What is the use of it? For example, generally we fix uh, one error and we estimate another error. What is that fixed error? What is the other error we are going to estimate? What is parametric test? What is the definition of level of significance? What is power of test? And what is the relation between the type 1 and type 2 error? And at what situation type 1 error is more important? And what situation type 2 error is more important? And what situation type 3 error is important? And these aspects we should know for the examination. Then some are easy traps questions. Like some questions looks very easy, but there are actually a trap. For example, you see the purpose of hybridization plant breeding. Generally, these options we can easily guess, but we generally miss why maintenance of lines. Why is the need of hybridization for maintaining the line? Generally, we'll eliminate this option and we'll keep BCD was the right answer. There will be option BCD, but was it wrong? For example, you think how do you maintain the A line in the utilization of male sterility, like cytoplasmic male sterility, CGMS? How do you maintain the A line? By crossing with the B line only. There, the hybridization used for the maintaining of the A lines. So, maintenance of the lines is also one of the correct answer. That's why ABCD were the actually ABCD option was the correct option. Then another important aspect is quality breeding. Generally, we stick on to the quality parameters that present in the BDC book, but it's not like that. There are some important research going in present era, like low GI-RS. The GI means glycemic index, low glycemic index. It's very important for the diabetic patients, and which are the institutes are working and which are the important land races having very low GI. We should also remember these points and low phytate MAGE, which land races are having low phytate MAGE and what are the markers for identified for low phytate and what is the use of low phytate MAGE and like generally they use for fodder purpose and soft wheat. What is the use of soft wheat? Which are the land races are important for the soft wheat? Which are the alleles are very important for the softness in the wheat? It's very classical. Alleles, their genes were also cloned for the softness of the wheat. And maize for the bioethanol production, which are the important quality parameters important for the increase the ethanol production. Then low rancidity per millet, low rancidity soybean, and gluten mimic traits in millets, and biofortification effects. There is an important document present published by the ICR, the biofortification efforts in the different crops by the Firoz Hassan. If you read this document, it's very, very important for the examination. Then another important aspect that you should read about PPFRA and patent. And you should clearly know what is the difference between PPFRA and patent. And you should also know that why India has developed its own mechanism of parental protection, unless it can maybe it could have followed existing rules like POVA rules are there, some patent rules are there. Why India has specifically chosen a separate mechanism for the protection of the varieties? These are the important aspects in the Indian scenario and plant breeding scenario that you should know about it. And definitely there will be a 2 to 3 questions will be from the these concepts. Then another important concept called as heterosis and fixation of heterosis. Recently they were asked about baby boom gene. There is question about MIME means not meiosis, not mitosis, meiosis gene. There are some important genes are present where synthetic apomixis and there are several important new developments are happening in the fixation of heterosis and several exceptions. For example, any theories which explain the heterosis were not explaining the concept of progressive heterosis and inbreeding, rapid decrease in the vigor. You should know what is progressive heterosis. It's simple concept like when in the case of autotetraploid, where double cross hybrids were more heterotic than the single cross hybrids. But in the case of allotetraploid, this concept will not apply. And it is exception for which theory of heterosis. You should know all these things, and these things are present in the one book called as Heterosis Reading by P.K. Mukherjee. Please refer that book for if you are interested to read these concepts. Then another important thing we should know about is different designs for the multi-evaluation trials. Nowadays, everyone are going for multi-evaluation trials and to estimate GEP interaction or to identify the better genotypes across the different locations or to test the adaptations. For example, in the initial stage of the parietal trial, if you're using RCBD, it's not possible because we can't. There will be large number of entries will be there. You can't use those designs which require a lot of resources. That type which design is most suitable, augmented designs and pre-rep design is partial replication designs and alpha lattice designs. And you should also know that what is the error degrees of freedom and total degrees of freedom in these designs and basic fundamental concepts about alpha lattice designs, pre-rep designs, and augmented designs, RCBD designs, LSD designs, flip-flop designs are very, very important for the examination. And another important called as sparse testing. It is also and what is genome relationship matrix and how genome relationship matrix were used in the sparse testing. What is the use of sparse testing in the large scale trials? And if you just type this word in the ChatGPT or in YouTube, you'll get so many videos that I'm not going to in detail about this concept. If you are interested, you can read. And these are the important concepts that I wanted to discuss today. And another important thing which you should remember is the Padma Shri awardees of this year. Like two scientists like Premlal Gautam and A.K. Singh sir actually awarded with Padma Shri for their contribution into the agriculture. And P.L. Gautam was Dr. P.L. Gautam sir was is the ex-chairman of former chairman of our Biodiversity Authority and as well as PPFRA. And Dr. A.K. Singh sir was the former director of IRA, and he released many basmati varieties that are worth about nowadays like 50,000 crores foreign revenue are generated by these varieties. And you should also read very important contribution of these scientists are important for the exam. These are the important for the examination. Okay, with this I conclude my presentation and all the best for your examination. Thank you. Nanda.